nothing new. The most important ingredient is, of course, the glucose. And I guess the only thing I'll emphasize for the glucose is, of course, we make sure there's plenty. We didn't skip on the glucose because that's like the most critical part of the experiment. We want plenty of glucose. We want them to have plenty to ferment or to undergo the aerobic respiration pathway. There is technically some peptone. It says digested casein, which is basically peptone. And usually we put plenty of peptone because we just want them to grow like crazy. We have a little bit of a problem here though, because one of the things we're going to look for is even the slightly acidic products from the aerobic respiration. So we actually had to just put a small amount here. Because you guys already know that usually when our organisms eat the peptone, it makes the pH more basic. So we don't want that to happen too much. This is to kind of prevent the pH from rising too much. We don't want to overpower the acids that we're trying to pick up from our pathway, so we just want a little bit, just so they have enough nitrogen so that they can grow. We actually don't want too much here. And then also, we have bromethymol blue. But what's really good about bromethymol blue is we've used it once already, which helps. We used it just last week on the Hectoin agar. That was the one we had the pretty colors. And bromethymol blue was the one where if it was really acidic, you kind of got like that bright orangey yellow. On your Petri dish, that was the one that E. coli turned the pretty orangey yellow. And then this is the one where if it was neutral, it stayed green. And if it became basic, it turned deep blue. So on the Hectoin agar, remember we looked for nothing to happen. So we actually looked for green or blue. On today's lab, though, our intention is we're going to focus in on the acidic products that are generated. So we're going to focus in on the orange. And then lastly, when you stabbed it, your media was not liquid and it wasn't solid. It was semi-solid because there was a small percentage of agar. Not the full percentage, just a small percentage. The reason why is because technically this is a motility test also. So you guys did the 2 stabs and we didn't put the full amount of agar in there so they can swim around and hopefully it'll allow for movement. So we're going to look at that. If your organisms are motile, when you check out your stabs, if your stabs look like they kind of got like a little cloudy or turbid, they kind of whitened out a little bit, and usually it gets kind of hazy. If that happened to your stab lines, then that's usually an indication that your organisms did move around. They were able to swim around, and so they do have flagella. So that's why the media was that consistency. It was kind of a softer consistency. Okay, number 4 though, that's not really like the main, it's a motility test built in, but the main kind of piece of information that we're going to gather is the pathway usage. So let's list the 3 most common results. What makes this lab very different from any other lab that we've done is you actually have to look at both test tubes in order to get your results. So you kind of need them both together. And then remember on one of the test tubes, we added the oil. Oops, I should probably make it a color other than green. We added The one with the oil or without the mineral oil? With the oil, right? So this is our F for fermentation. Fermentation is anaerobic, so we're going to rely on the test tube with the oil. This is our O test tube. So that's why the lab manual calls us the OF test. Oxidation fermentation test. And so one of the more popular results, remember when you guys stabbed, everything was green. We started off with that forest green color. So usually one thing that can happen is you get the green with kind of the brighter yellowy orange on top. All right, so usually what I like to do is I like to look at the F test tube first. You look at your F test tube, totally stays green, nothing happens in there. Are you positive or negative for fermentation? Negative, right? So stays neutral, like there's not really a shift. Basically what happened was you added the mineral oil on there, and it looks like once you added that oil on, the organism couldn't grow at all. We didn't even get blue, we didn't get anything. It just stayed green. So your organism didn't grow. It's negative. It definitely didn't use the glucose. Because if it had fermented the glucose, we would have expected some organic acid production in there. In our O test tube, though, we do have some signs of some acid production. So, and since we know our organism can't ferment, because in our F test tube, there's no signs of fermentation at all. We know it's not fermentation. And this test tube has plenty of oxygen. So the only thing we can conclude that's going on the left is it must be using that other pathway, that aerobic respiration pathway. It's definitely not fermentation because once you add that mineral oil, organism just does not thrive at all. So since it only metabolizes when there's oxygen around, bless you, it must be that this has to be an obligate aerobe because it's totally reliant on oxygen. Oxygen is required for usage of glucose. So glucose cannot be metabolized if oxygen is not around. You have to have oxygen present in order for this particular organism to metabolize the glucose. So that's one popular result that a lot of people will get. The other possibility and this is our, you guys added some oil. We said this is our F, this is our O. So we're actually getting two results because we have our two separate test tubes. So another popular result that a lot of times people look at is maybe a little bit of green. And then the rest of the test tube. You get some nice, bright, pretty colors here with the bromthymol sprinkled in. All right. So we check out our F test tube, which is the fermentation test tube. And are we positive or negative there? Definitely positive, right? We definitely have some acid. We have the oil on there, so in that test tube with the oil, the only possibility is fermentation, that's it. So it's definitely positive for fermentation. And then what about our O test tube? We have some acid there, so are we positive there? Yeah. So we say, okay. And then this is the test tube with the oxygen around, right? So definitely, like one of the possibilities is, yeah, it's using that other pathway, oxidation. We have some oxidation going on. So we kind of get the double positive, where organisms, if oxygen is around, they'll use it for aerobic respiration. If you cut it off, then they'll switch into their fermentation mode. And in lecture, we actually call these facultative anaerobes. In lecture, though, we said really like the ideal is with the oxygen, right? Because they're kind of, that's their preference, and they kind of grow a little bit better. But if you cut off the oxygen, it's not like they're going to die. They'll still be able to ferment the glucose and grow in the incubator just fine. There is another possibility, though. So for sure, this is fermentation going on in here. The other possibility, though, that we did mention in lecture is there are some organisms, it doesn't matter if the oxygen is cut off or if the oxygen is there, they just kind of ignore it, right? So we said there's this category called aerotolerance, where they never use oxygen either way. Even if it's there, they just kind of ignore it. They never metabolize it. It's never part of their pathway. So either way, what happens is they just ferment, no matter what. That's what makes them aerotolerant, is that no matter what, they're just going to ferment. So they never, ever use it. Now these two results are kind of lumped in together, but we are going to be able to differentiate them on today's lab. You guys are going to set up what's called the catalase test. And on the catalase test, you will be able to distinguish facultatives from aerotolerance. So, you know, they're kind of lumped in together right now, but we'll be able to split it up. And then lastly, really quickly, sometimes it happens where people will inoculate, and this is the test tube with the oil, this is the F, this is the O. Sometimes what happens is you just kind of get the green everywhere. With a little bit of blue on top. So for this category, I am going to kind of put this though up the side. Number 3 is not very common. It's definitely more rare for an organism to not use glucose at all, because glucose is such an essential sugar, but that is a possibility where you get like the double negative, no fermentation, no oxidation, and oxygen requirements you can't really assess because it's like a double negative. So in that last result, there are some organisms out there where they can't use glucose at all. It's pretty rare though. They can't use glucose at all. So, but usually 1 and 2 are the more popular